Hey guys, welcome back. Season 1, Episode 2 of... I don't know, we gotta find a cool name for the podcast. I'm gonna get back to you on that one. This is actually take 3 because once again, I'm coming to you live from a Mythic Plus dungeon. And the first 2 dungeons I tried, oof, they were rough. So we're gonna give it third time the charm here. I had to do... This is my third take because the first 2, literally, it was just the key was so bad, it was so hard that, yeah, I couldn't even get through a full thought without freaking out. Anyway, okay, what I wanted to, actually first, good reviews. I've heard like 2 reviews. 100% of those reviews were good. Alright, so we're at a 100% good review rate, 2 out of 2 on the episode 1. And kind of, you know, there's some things I'll work on and stuff. I don't know where this is going. Who knows? We're just doing it for fun. I did get some feedback that said perhaps you should, let people know of your qualifications, so you're not just some scrub reading things from Wikipedia or something. And I think that's fair. I don't want this to be about me at all, though. This is probably like the one and only time I'll even just mention this stuff. I got my undergrad in chemistry. Undergraduate in chemistry, and then I did a master's in polymer chemistry, where I focused on polymer nanoparticles for cancer therapy. And right now I am in my 4th year of my PhD in pharmaceutical chemistry. So all the fields are sort of roughly related: chemistry, polymer chemistry, and then pharmaceutical chemistry is what I'll do the PhD in. And we're almost done. We're just about wrapping it up. I've published a bunch of papers, 3 papers, and I'm writing a 4th one right now. All kind of in the area of cancer research, so that's where we're at. That's the first and last time I'll probably ever mention that. So yeah, it's not about me. Who cares? So it's moving on. Today I wanted to talk about actually something very, very cool, and I feel bad that it's sort of became this sort of, I don't even know, polarized word, but mRNA vaccines. I'm not here to convince anybody of anything. Do you? Half my family has been vaccinated, the other half hasn't been vaccinated. I'm in the middle. I'm Switzerland. I don't have, I myself was vaccinated and boosted. I don't have an opinion one side or the other. You do you. We'll just talk about what mRNA is and what it brings to the field as a potential therapeutic, because it's really cool. Really cool. This is just the tip of the iceberg. This is the first FDA approved mRNA product, and there's only more. It's just more coming. So it's like the tip of the spear here. But what is it? Low key, what is mRNA? That word's thrown around a lot, and I think like in my world it's not a new concept at all. We talk about mRNAs all the time, like it's just such a common thing, but as a therapeutic it hasn't been possible until very recently, and that's due to a number of reasons. But so as it's kind of new in the public's eyes, there's sort of, not a lot of people know exactly what it is. There's a lot, I don't want to say like any ill-willed people are going around spreading false information. I think there's just a lot of, poor information or just incorrect information that's getting spread around and propagated. And then that leads to fear, and that leads to distrust, and that leads to... So I'm just going to do my role. Let's just talk about what mRNA is. You can decide for yourself anything beyond that. Let's at least just establish a baseline of what mRNA is, because it's actually a really cool therapeutic. And I'm going to be doing this while healing. As a discipline priest in the game World of Warcraft in Mythic Plus Dungeon. So off we go. And as I was trying to think through how I would present or start off with this topic, I think a discussion about the immune system is pretty necessary. Just so we can establish a baseline with just a few simple concepts of the immune system, what it is, what we're trying to achieve with therapeutics that, vaccines in general. What are we trying to do? What's the point? Who cares? Well, they're actually a really, really cool technology. So let's try to understand a little bit about it. So the immune system, very, very complicated. We are not here to get into all the intricacies of the immune system. What we can talk about, I don't know. So we're trying to do a weird skip here in the game, and I don't know if the warlock's having problems with it or. Anyway, I'm gonna continue on. So, lots of different cell types in the immune system. What the immune system is kind of how I picture it. It's a blank canvas. When we're born, blank canvas. We need to protect our bodies from the world. And if you can, that seems like such a huge, monumental task. How in the heck do we do that? How do we fend off all the bacterias, all the viruses, all the fungi, all the parasites? How do we protect ourselves from all that's out there? Well, we have two general methods as human beings, and that is innate immunity and adaptive immunity. And we're just going to talk about our adaptive immunity today, which means we have part of our immune system that can literally adapt to its surroundings and to what's trying to infect it. It can learn, it can adapt, it has memory. That's obviously pretty interesting, pretty cool. So when we vaccinate somebody, we're trying to tap into this memory part, this adaptive immunity. Let's just talk about kind of how that works. In order to understand how that works, we need to understand a few cell types. So there's 3 cell types, I think, that we can break it down into that are most important for our discussion. There's B cells, there's T cells, and then what we call antigen presenting cells. Kind of a weird word. In my world we use it all the time, but I know that's probably not a very common word for everyone else. But an antigen is something that a cell can bind to, or something that a receptor can recognize. Could be a protein, could be a sugar, could be some kind of structural conformation, but it's just the word we call the thing that we are targeting, or the thing that we're trying to bind. That's called the antigen. And so an antigen presenting cell is a cell in our immune system that its whole goal, its whole role is to just take up the virus particles, take up the bacteria cell, or take up an infected human cell. And then when it's taken it up, it breaks it into a bunch of pieces. And then it takes kind of like a Christmas tree almost. That's how I picture it in my head. It takes all those pieces and then decorates its surface, kind of like the ornaments on a Christmas tree. And we'll talk about why that's important in a second. So those are the 3 cell types that we'll talk about just for today. So first, a B cell. These are the cells that create our antibodies. And what's so cool, B cells are part of our adaptive immunity, which means they have the ability to, we call it like learning. They can learn what to attack. And they do this by, in their development stage, they create these proteins that they put on their surface. Any protein that a cell puts on a surface, we typically call it a receptor. So they make these receptors that bind one antigen, and every B cell is completely different. It binds a completely different antigen. And there's, who knows, right? Trillions of B cells in our body. And so if you can imagine, we can potentially bind trillions of different antigens. Now most, 99.99999% of our B cells will never see their antigen. Our B cells will be created, never see their antigen, so they'll just die. But on the off chance that our B cell recognizes the thing it was made to recognize, sort of randomly, just kind of a shotgun approach. Let's call it COVID. It recognizes, oh, I randomly made this protein that binds to COVID. Okay, I found the thing that I was looking for. I found the thing I was made for. Let me make a bunch of copies of myself. And so I'm going to make trillions of copies of myself. And now I have a whole army of B cells that recognize the same thing. And that's how adaptive immunity works. So that's a B cell. T cells work in a very, very similar fashion. They don't make antibodies, but they do make unique receptors, just like a B cell. And their function's a little different. So a B cell typically makes antibodies, and they can flag infected cells, or they can flag the virus where they just bind to the virus, or they can even take up the virus. But a T cell's different. A T cell is the workhorse. A T cell is made to kill infected cells. That's what a T cell does. It recognizes. We just messed up a small mechanic right there. A T cell will recognize an infected cell based on what that cell is sort of communicating to it. Like, hey, I'm infected. I'm infected with this thing. You need to kill me. So a T cell is really good at killing cells. And it has that same adaptive immunity, so it can sort of learn what to look for. Now, an antigen presenting cell, their whole role, kind of what we talked about earlier, is to wow, I've never seen this pulled with this pull. This is new for me here. No! I died. I thought I was out of that mechanic there. That's sad. Yeah, okay. Oh, our tank just died too. Oh no. The wheels are coming off. It's gonna hit me again. Not like this. Okay. Wait. Oh, our tank's alive down here. Where am I? Just laid on hands. Okay, wow. The wheels came off harder than ever right there. That was all because I died, so we can put that one on me. Okay, back to it. So antigen presenting cells, their whole goal, if you remember, they just take up viruses or bacteria cells or infected human cells, break them apart, get all their different pieces, and just put them on the surface. And that, so now you can kind of understand what that's doing. Now, it's showing the B and T cells what to look for. Hey, I found this thing in the body, be on the lookout for this stuff. And so if a B cell or a T cell has a receptor that can recognize the stuff that's on the surface, they will multiply, make more of the copies of themselves, and you can mount an immune system. Okay, so now that baseline stuff, so now we sort of have a basic understanding of what adaptive immunity is. With a vaccine, all vaccines are the exact same. What we're trying to do is like show the blank canvas, like, hey, look for this thing. And, you know, before this thing gets in your body, you need to be on the lookout for this. Dude, this is some wonky pulling. I gotta keep my head on a swivel here. So every single vaccine is made with that in mind. We are trying to get our B and T cells prepped for some kind of foreign invader or a bug that's going around society, whether it's the flu or measles, smallpox, whatever. We inject our bodies with small pieces so that our B and T cells can learn what to look for before the real thing gets into our body. And so we don't see the entire virus, right? Like there's a lot of the virus we don't see, but when we get vaccinated, I'm talking about, there's a lot of the virus. It's not the real thing, so we don't see everything, but we see some parts of it, and so we can be a little bit better prepared rather than just a blank canvas, you know, uh, COVID getting into someone who's never been vaccinated. It's just a blank canvas. Their immune system has never seen any components, so they don't really know what's going on. It's just going to take the immune system a little bit longer time to figure out, you know, what's going on, figure out how to get the, wow, kind of messed up bad there. Figure out which B cells match, which T cells match up, but it'll still happen. You know, it's not like it's not going to happen. It's still going to happen. But the whole idea of a vaccine is we want it to happen fast, so we have better recovery rates, we have less symptoms. That's the whole idea. So how we do it as a therapeutic then, typically we take, what's been done in the past, until this mRNA stuff, is we take small pieces of, let's just use a virus for an example. We just take small pieces of a virus that we're worried is spreading through the population, and we put it into basically a shot form, and then we inject that into a muscle. And we use the muscle because there's lots of these antigen presenting cells just sort of stationed throughout our muscles, just ready, kind of just waiting, ready. So we inject the stuff right to our antigen presenting cells. They immediately take it up, break it apart, and present it on the surface. And then it's communicating with the B and T cells, hey, look for this stuff. Who can match up with this? Someone match up with this. And then B and T cells will, you know, kind of scan and figure out if they can bind. And the stuff that can bind, the cells that can bind, they will multiply. And then we have this sort of memory, immune cell memory, where some of those cells are going to stay alive forever. We call them memory cells. So that if in the future, 2 years from now, if we happen to randomly get this virus, we'll have some memory cells that have already been made that match up with this virus. So we inject into our muscle. What is this pole? This ain't it, dude. I mean, this is it, but I don't know if this is it for when I'm trying to do a damn podcast here. Oh, almost just got kicked on a really bad time. Oh yeah, I was actually insane. Okay. Okay, sorry, losing my train of thought because that was insane. That was literally insane. I loved it, but not when I'm trying to film a podcast. Okay. Holy hell. Alright. So we inject the vaccine into the muscle. Antigen presenting cells pick up all the stuff that we want our immune cells to be looking out for. Presents them on their surface. B and T cells match up. They say, hey, I recognize that thing. Let me make a bunch of copies of myself. Let me make some memory cells so I can remember this thing forever. Okay, so when, you know, we realized, I think like in the late 1800s, when people were doing research on smallpox, they sort of realized the maids who took care of cows, milked the cows and, you know, whatever, worked with the cows, the maids seemed to be immune to smallpox. And after some investigation, what they realized is there was a cowpox, slightly different strain, related but different. It was a smallpox of cows, so it infected cows. A little bit different. It didn't infect humans. Well, it could infect humans, but it wasn't bad. It wasn't deadly in humans. And these milkmaids who were working with the cows seemed to be immune to smallpox. And so they sort of realized, wait a second, I can just expose people or inoculate people with cowpox and they will be immune. Oh no. And they will be immune to the actual smallpox. So I think that's the first reported sort of, you know, successful, it was clear back in the 1800s, successful use of a vaccine. And it was very primitive in those days. You know, I think they literally just took the pus from the sores on cows. They took that pus and like wiped it on people, and that was their vaccine. So kind of sketch, but it worked. And that's kind of how the science of vaccines developed, where we realized we can, give even like a lesser version or a weaker version of what we want to be immune to a person, and that person will be immune next time they get in contact with the real thing. And immune's a strong word. I shouldn't say immune. They can still get infected, but it won't be as severe. They won't have as severe side effects. They'll have much less chance to die because their immune system has already seen it. It's already had time to mount a response. That's the whole idea. So up until this mRNA vaccine, what we've gotten in the past, what I got as a kid, mostly I'd say unless you're like an anti-vax type person who just had never got a vaccine, everyone's been vaccinated for something. For the most part, as kids we all got vaccines and stuff, and that's for diseases. We've literally wiped diseases off the face of the earth due to vaccines. And up to just a few years ago, the only vaccines, so what we were putting into people, pieces that we were putting from the virus or whatever we were trying to inoculate against, was protein. We would just take little bits of protein from the virus, say, and inject it into the person. And then our antigen presenting cells would pick up that protein, get it on its surface. B and T cells will then take all the time it needs to match up, get all their matching receptors, and then multiply. And then they could have memory cells, and then we are vaccinated against that virus for many years. And so that's the thing, is the memory cells some live forever. You just have them for life, but most of them don't. That's why you need to get revaccinated for some stuff like every 10 years, because you lose your memory B cells over time, or your memory T cells over time. So you just get revaccinated, and the same thing happens. You create antibodies and you go through the whole thing. Now, what's really cool about... Okay, so there's two options. You can break up pieces and you can inoculate with just protein fragments. Or you can actually take what we called a live attenuated vaccine, where they've actually taken a live strain of the virus we're trying to fight. They grow it in a petri dish, infect cells over and over and over and over, and many generations later, they find a version that's very weak. It has some activity, but it's very, very weak. And then it has a lot of the same proteins and a lot of the same structures, so we can then inject that into a person. And so it's like a very weakened form of the real thing. That's called a live attenuated, but honestly those aren't even... I don't even know if they're used anymore. Those were a thing a while ago. They might be used for the flu, actually. The flu vaccine might be live attenuated. Just like a very weakened form that has a lot of the same proteins. We actually timed that key and did really well. I haven't even gotten to mRNAs. It only took us 19 minutes. I was thinking that would take a little longer. Okay. So, another thing they can do, instead of a live attenuated, they can take the virus and irradiate it with radiation. Basically like cross-links. It kind of just deactivates, kills the virus. And then they can put that sort of like dead version of the virus. They can inoculate you with that. Now for mRNA, it's a little bit different. So if you remember back from like junior high biology, you have DNA that codes for all of your genes. DNA gets transcribed into RNA. RNA can then leave the nucleus. And that type of RNA that leaves the nucleus, that's called mRNA. That's where that comes from. That's messenger RNA. And messenger RNA leaves the nucleus, goes into the cytosol, or it goes into, I don't know what else to call it. Just kind of like the cell fluid, kind of in the middle of no organelles, just kind of in the fluid of the cell, where it gets translated. The RNA codes for a protein. So it gets translated into a protein. That's the basic fundamental dogma of living cells. DNA to RNA to protein. Protein then does function, does everything that a cell needs to do. So there's hundreds of thousands of different proteins. Yeah. DNA to RNA to protein. So what we're doing, instead of using protein, using the end piece to inoculate with, now in mRNA vaccines, we're using the mRNA component. So we take the mRNA. Let me just try to find a new key here, because we'll just keep this party rolling. So we take the mRNA that codes for the protein that we want to inoculate against. So we're not using the mRNA to get our T and B cells to recognize the mRNA. That's not the point. We're trying to get an antigen-presenting cell to take up the mRNA. And while the mRNA is in the antigen-presenting cell cytosol, it's going to turn it, it's going to read the code and turn it into a protein. And that protein is going to be a viral protein. In the case of COVID, it's the spike protein. So our own cells, our macrophages, our dendritic cells, they are making viral spike protein in their cytosol. And then that gets chewed up, and then that gets presented on the surface. And so little pieces of the spike protein are presented all over the surface, and then B and T cells will sort of match up and see what B and T cells recognize, which ones recognize, they will multiply. That's the entire idea. And what's so cool about the delivery method, so mRNA is a form of genetic code, basically. It's a code. And our body is on high alert at all times to look for foreign code and degrade it immediately. So if we put mRNA, if we inject mRNA into the blood, I actually didn't even get invited to that key. That's kind of odd. So if we just took mRNA in solution, in saline, and then injected it into our arm, it would do nothing, because it would get degraded like that fast. It would just degrade instantly. It wouldn't be able to do anything. Our body is so on the lookout for foreign genetic material that it would just degrade it instantly. Yeah, so mRNA is not very stable. It'll get degraded. Our enzymes in our body get cleared from the blood immediately. So up until just a few years ago, like, we didn't know how to make it a therapeutic. We realized, yeah, we can use this as coding technology, but how do we deliver it? We can't get it to, it's not stable. How do we use it as a therapeutic? Doesn't make any sense. Well, you need to be able to package it into something. So I debated, should I do this episode on nanotechnology or should I do it on vaccines? I chose vaccines first. The next episode is going to be on nanotechnology, which is going to talk more about how the mRNA gets packaged up. But basically what we need to do is we need to conceal the mRNA from the body. We need to protect it. Because if we just put the mRNA in the body, it's gone. It's not a good therapeutic. Not going to work. So we put the mRNA in little vesicles, little fat droplets called liposomes, or lipid nanoparticles. A lot of terminology there. We'll talk about that in the next episode, but I just got called sir. Hello, sir. Anyway, so we package the mRNA up into these little, almost like, you can just think of it like a fat droplet. And now it's hidden from the body. The body doesn't know there's mRNA inside that fat droplet. And so that's what we're injecting into our arm. Basically these lipids that are just encapsulating our mRNA, we call something that's encapsulated like that a cargo. It's kind of a cool word. So it's encapsulating our cargo, our mRNA, protecting it from the harsh degradation environment of the body. And then so what our antigen presenting cells are then taking up are these fat droplets. Breaking apart the fat droplets. Oh, look, there's mRNA in here. It's a code, so it actually, we kind of like trick the cells. The cells just recognize it as mRNA. They scan it, they make a protein out of it, and it's the spike protein. It's really, really cool. You can imagine then, okay, we don't have to stop at just the spike protein. We can do any protein. We can use the mRNA code for any protein. As long as we know the sequence of amino acids of our target protein, we can make the mRNA code for that. So the sky's the limit, right? We could do anything. We could do any protein we want to vaccinate against. And that's why it's such a big... We don't need the virus anymore. We don't need a live attenuated virus. We don't need to irradiate the virus to kill it. We don't need to harvest proteins from the virus. We just analyze the amino acid sequence of the protein we want to vaccinate against, and then we can make our mRNA code. Package it up into a fat droplet, inject it into someone's arm. Boom. We can vaccinate against anything. That's the theory. That's why it's such a cool technology. And what's nice about it is it's a platform technology, right? We don't need to change anything about the fat droplet. That all can stay the same. The only thing that changes is the mRNA code. So you just plug in the code that you want into the same system, and we can vaccinate against anything. You don't need to make a new one every single time. You know, it's just plug and play. Really simplifies, streamlines the process, and that's how they were able to get, you know, what is it, 4 or 5 different vaccines approved in like a year. As soon as they saw the sequence of COVID, they saw the entire genomic sequence, they knew exactly what the spike protein sequence was, they knew the code. Okay, we're just going to borrow this code and make our own mRNA, and we're just going to use it as a vaccine. Simple as that. My tank's getting absolutely obliterated. First pull. So let's talk about, so that's mRNA. So mRNA is the code that we're sort of tricking our antigen presenting cells, thinking it's their own mRNA, and so they turn it into a protein. Risks. What are some risks? Well, inherently, everyone's immune system is completely different, right? That's the nature of it. Everyone's B cells are different from everyone else's. Everyone's T cells are different from everyone else's. And so we cannot standardize what antibodies are going to get made in people. Your antibodies are going to be different than my antibodies. Your B cells are going to be different than my B cells. And there's no way around that. That's just always going to be the case. And that's not necessarily a bad thing, but how it can be bad is when the this is very rare, just want to preface with that. When the antigen presenting cell is tearing apart the fat droplet, there's other components besides the fat and the mRNA. You know, we call them adjuvants, but they are things that we've put in there strategically to sort of kickstart the antigen presenting cells to, you know, engulf what we want it to engulf. We're sort of kickstarting the whole process. And so that also gets put onto the surface of the antigen presenting cell. And very rarely, your B and T cells will actually make antibodies against that. So, and that's, then you have an allergic reaction to the delivery vehicle. Usually the first time you have a little bit of a warning. You'll get probably like, you know, some small inflammation and stuff. Very, very rarely it can be more severe. So allergic reactions are by far the most common negative side effect of these things. Of vaccines in general. Not just COVID vaccine, but just all vaccines. Yeah. And then, you know, I don't want to get, we're not going to get too down that road because there's a lot of, I understand there's a lot of emotionally charged points of view, and for good reason, and we don't need to get into it at all, but just, yeah. So just in general, that's why when you go in to get like the flu shot or something, they ask you if you have any allergies to eggs. It's because some of the adjuvant they put in there is related to, is it the yolk? I'm not exactly sure. Some part of the egg. So if you have a pre-existing allergy to it, yeah, they want to know that because they're not gonna, your antigen presenting cells are gonna put that protein on their surface, and it's gonna aggravate your B and T cells that respond to the egg. Yeah, so that's mRNA, and that's vaccines. I don't know. We can definitely go into way more detail than that, but I think that's a pretty good level of understanding for, like, I don't know, general conversation in everyday life. That's probably like the extent. Actually a really cool technology, the mRNA vaccines, because it's a platform, right? We can plug in whatever mRNA we want. We can then code for whatever protein we want, protect people against many different, infinite amounts of different proteins. So that's why it's such a big idea, cool idea in the science realm. Now I realize there's lots of different points of view. So just, yeah, if you stayed this long, I appreciate it. I assume it's probably not, it's definitely probably like a very polarizing topic. Hopefully I didn't offend anybody. We're just looking at it. I try to be as objective as possible. Sensitive subject, I understand. And it shouldn't be, honestly, but it is, unfortunately. But, yeah. Thanks for listening. I will do the next episode on nanotechnology, which is my specialty. That's my field. And it's kind of funny, like I hear that term nanotechnology thrown around a lot, sort of. It's almost like pop culture now. Lots of headlines, lots of news stories. You know, some new nanotechnology at MIT that's going to change the world. So, talk about what that means in the next episode. Again, thanks for watching, and thanks for any support. Give me some feedback too. I'd love any feedback, and I'll see you next time.